Great, thank you very much for the opportunity to present our work here today. Whoa, that's not the first slide. There we go. So we're all here for the same reasons. I'll keep the introduction brief. Anne already very nicely introduced the value of a sort of a high-dimensional readout to characterize variants. And so what we set out to do is really how do we develop a method that's both very scalable and provides not just information on the pathogenicity of a variant, but also its function and enables mechanistic interpretation. And so one way to do this is to profile variant effects using a single-cell sequencing readout. And the way we develop this method is essentially a fairly standard lentiviral overexpression assay where we tag each variant in a saturation with the genesis library with a random barcode. And this barcode is read out directly out of the standard single-cell sequencing. But then we have a second step where we link a random barcode to the variant in each cell by using amplicon sequencing. So just tiling the region of the gene and then doing variant calling on the resulting pileups for each gene. And we validated this using long-read sequencing from PacBio, obtaining extremely high concordance rate between the two approaches. And so I just want to briefly summarize the approach in general and work with recently published in a preprint characterizing 3 genes. So we looked at TP53, P16, and SOD1. And what I'm illustrating on this slide here is essentially how this data looks like. So I'm not showing the single cell profiles because these are generally very noisy. Because of cost limitations, essentially, we can profile thousands of cells per variant. So we typically end up with about 20 to 100 cells, somewhere in that ballpark per variant to get reasonable signal. And so what I'm showing you here is a UMAP projection of the average profiles per variant for TP53. And you can sort of see that, and the labels here are computational predictions of pathogenicity from the primate AI method. And what you can see is that there's a sort of central cluster of variants that corresponds to where the synonymous variants lie, but then also variants in smaller clusters here on the bottom left, as well as these variants that are predicted to be pathogenic in this cluster here. And the data at the level of individual variants is still relatively noisy. So then we have, depending on the gene, we perform the averages slightly differently, but overall what we generally do is we take the structure predictions from AlphaFold and do averaging in small neighborhoods around residues. And for TP53, this then enables us to get a much more detailed view of the gene with clearly distinct clusters, notably with variant effects that correspond to the DNA binding domain and the transactivation domain. And so this was very encouraging to see that we're actually not just getting a relatively good separation of pathogenic from synonymous variants, but also seeing clearly domain-specific effects. And we've benchmarked this. I won't go into the details today, but you're welcome to check out the preprints against existing experimental scores for the genes we looked at, as well as a wide range of state-of-the-art computational predictors, and showed that the expression, sort of the single-cell expression-derived scores systematically sort of outperform the other predictors. And so what I'd like to do today is give you a sort of a glimpse into the results that we've obtained for 2 new genes we've looked at. So SCHB, which is a tumor suppressor with functions in the citric acid cycle, as well as oxidative phosphorylation pathways, and where germline mutations underlie endocrine tumors and gastrointestinal stromal tumors, as well as SRSF2, which is splicing factor, part of the spliceosome machinery, where mutations that a single residue are commonly found, and those underlie a typical CML as well as ALL. And so very briefly, in terms of SDHP function, the only thing that you really need to know for the talk today is that SDHP is part of a complex with 3 other subunits, sort of like here in the middle. And crucially, it holds these 3 iron-sulfur clusters that are responsible for electron transport throughout the complex. And so we focused on SCHB and sequenced about 70,000 cells, expressing about 2,400 of all possible single nucleotide variants in this gene. And we're now going to look at the effects of these variants, both on just looking at proliferation. So essentially, we can do the same thing you would do in sort of a one-dimensional readout, just looking at the abundance of variants we observe in cells over their abundance in the initial library. And then, in addition to that, we can take the transcriptomic profiles and calculate predictions of pathogenicity based on those scores. And so typically we visualize our results using these kind of representations. So at the top here, I'm showing you what the average transcriptome profile across all amino acid positions in the gene looks like. So with the transcriptome represented as Z-scores, sort of variants that increase expression shown in red, variants that decrease it shown in blue, with the proliferation score at the bottom, where we can use synonymous variants to calculate which variants are significantly depleted or enriched relative to this, and apply FDR, which shows sort of effects consistent with the domain-specific effects we see in the expression, and then below that our expression score, which is also highly consistent with the depletion observed in proliferation. And in the bottom track, the predictions from primate AI, where I've also labeled the variants annotated in ClinVar, whether they're pathogenic or benign. And so there's a very nice concordance here between these three scores and the location of the ClinVar variants, notably enriched in the two domains that hold these iron-sulfur clusters that I mentioned. And overall, the effects on the transcriptome that we observe are consistent with the known function of the gene. So I've highlighted a couple of the pathways here, including hypoxia, reactive oxygen species, and metabolic pathways. We can then always, of course, we want to benchmark the performance of our scores. And this can be quite challenging in some cases, especially because of the very small number of benign variants that are often annotated in ClinVar. And so what we're doing is we take variants that we observe at moderately common but still quite rare frequencies observed in UK Biobank, NOMAD, TopMed, et cetera, and to complement putatively benign variants in this benchmark. And similar to the previous genes we characterized, we observed, essentially we obtained a perfect classifiers for these variants based on the expression score, but also very good performance with the other predictors here, including computational predictors from primate AI. One thing to note is that the correlation that I'm showing here between the predictions from primate AI and the expression score from our assay is that the correlation itself is not very high, and there's notably a gradient of the expression score that's also strongly correlated with proliferation. We're still looking into this and trying to understand this, but it's certainly promising that we get this sort of non-binary readout that you would get with a certain classification. But the challenge here is really the absence of good benchmarking data sets to compare against and understand this better. So next we wanted to understand a little bit better what's going on functionally with the positions that we see depleted in our results. And so when we label the different scores we have directly on the structure, we can see that there's sort of nice enrichment around certain position, notably in the vicinity of where the iron-sulfur clusters are. And we tested this explicitly, sort of asking whether the enrichment we see, what the probability of this would be by chance, given the total number of mutated residues that we observe. And indeed, this suggests that there's a strong enrichment of mutations near each of the positions of where these iron-sulfur clusters are. And we can turn this around, and instead of just focusing on these iron-sulfur clusters, we can ask at every position in the structure, what's the probability of seeing a non-random enrichment of residues with a significant effect at that location. And that's what I'm illustrating in the track here at the bottom. This thing is hard to point sometimes. Anyways, so for most of these positions that are significant, we see that they indeed fall again where we see where the iron-sulfur clusters are located. But in some other cases, they actually appear to just disrupt binding between the different subunits of SCHB. And in the case of this gene, actually, no matter where the variant is, the functional consequences, at least in our assay, seem to be uniformly the same. So we see consistently for all the variants that show a significant effect, the same effect on the transcriptome. So next I want to talk a little bit about our results for SRSF2. Very similar visualization to before, where SRSF2 essentially has an RNA recognition domain and then a large sort of unstructured tail And I've mentioned before that the most commonly observed mutations are these P95 mutations. But very interestingly, in the assay, we now both see additional variants that seem to have similarly depleting effects, as well as variants that show increased proliferation. And so when we map this onto the structure, what we find is that the variants that have decreased proliferation actually sort of share the common properties that they're facing towards where SRSF2 binds RNA, and likely acting by either disrupting binding or altering motif recognition. And so since SRSF2 is a splicing factor, the natural question is to ask, can we actually detect any effects on splicing? And that's not something that's completely obvious, given that our readout is sort of based on 3' single-cell sequencing. But there's actually quite a bit of splicing information that you can recover. And here, just to illustrate, this is essentially the top panel that I'm showing here is the kind of coverage you get from RNA-seq from a bulk readout. So this is published data looking at SRSF2 variants in K562 cells. And you can nicely see that there's sort of even coverage across the exons of the gene that I'm showing here, as well as in red, plenty of splicing information linking the different exon-exon junctions. When we look at this in our own data, based on the single-cell readout, this is much more heterogeneous. But nonetheless, you can see that there's relatively decent coverage on some exons, and there's plenty of splicing information. And so now, instead of just taking, when we calculate our expression scores and build our predictors, instead of taking a vector of gene expression for every variant effect, we construct a similar vector using splicing ratios. And since this data is fairly heterogeneous and we only get coverage in certain portions of the genes, we apply a sort of really adapt what's been published in this method called leaf cutter, which looks at differences in splicing effects very locally. So it builds connected component clusters of junctions, and then we can compute how ratios between the junctions in those components vary across conditions. And here's how this looks for our data. So instead of now projecting the transcriptome, I'm now projecting the splicing ratios. And you can see that there's now clusters that are significantly distinct from how synonymous variants would look like. And there's a cluster that looks distinct that corresponds to variants that have a positive effect on proliferation, and then a separate cluster that corresponds to negative effects on proliferation. And you can also see that several of these variants that are depleted in completely different positions of the gene, actually also here in terms of where they project based on their splicing score, appear to be in vicinity to one another. And then we can take these the splicing profiles corresponding to these different clusters and now look at specific events. And I'll just highlight here at the bottom one of the most well-known consequences of the P95 mutation is actually increased splicing of a poison exon in the EZH2 gene. And what I'm showing in this plot is essentially just focusing on the two neighboring exons and comparing how the splicing profile that we get in K562 cells that carry a P95 mutant and our single-cell data. And we can essentially observe exactly the same thing. So that in both cases, despite having very few cells and very few reads that cover these junctions, we still see a significantly increased inclusion of this poison exon in the single-cell data. And then conversely now for some of the other clusters, since these have not been characterized before, we can look at which genes show the strongest splicing differences and then try to understand what the potential mechanism underlying this is. But that's something we're still working on and is quite challenging. But here's just one example corresponding to these variants that lead to increased proliferation, where we also see significant differences in splicing. So with that, I'll conclude, and I'll just summarize. I hope I have convinced you that there's really great promise in using single-cell sequencing as a readout for these types of assays and the rich phenotypic information that they enable, especially also the inclusion of splicing information and not just expression. I don't have time to show you this today, but we actually also see nice splicing effects in genes we characterized previously, including TP53. So happy to take any questions. Thank you very much for your attention.
Questions? See somebody up there in the back? Are you okay. Why don't you go first since the microphone is running? There's one.
Thank Thank you. Great talk. This is Jack Damaris from Cold Spring Harbor. So you were saying earlier that you averaged over variants that are close to each other in sequence to reduce the noise. If I could adjust the number of variants in my assays to make this work, like if I wanted to not average spatially, how many cells would you say that I would need per variant to get a confident call without doing like local structure averaging?
So this is obviously just sort of extrapolating from the number of cells that we need, but it's probably a few hundred. And that just because we're testing thousands of different SNPs, that just in terms of scalability and cost at the moment is not really feasible yet. I mean, there's a very nice early paper from Vivergeff's lab or from Ursu et al. That sort of pioneered using a single cell sequencing readout, and they actually needed about between 500 and 1,000 cells per variant to get decent signal. So I think if you want to focus on really individual variants, yeah, the ballpark is in the several hundred cells per variant.
Awesome. Thank you so much.
Okay, that is all we have time for. Thank you so much.